is mutation and paraganglioma in cyanotic congenital heart disease. Paragangliomas are very rare neuroendocrine tumors that originate in paraganglioma. They can affect people at any age, but the most frequent occur in patients between 30 and 50 years. These are usually benign and slow-growing tumors, but they can move to other near parts of the body where they become cancerous and can metastasize.
In various parts of the body. These cells are located in the middle ear, up against the inner ear, and also located in an area called the jugular bulb. And if I were to look back at the back part of this model, I would see that the sigmoid sinus, this is a major vein that drains blood from the brain and empties into a large channel called the jugular bulb. This also has the same sort of cells that we're talking about in the middle ear, the cells called paraganglia. Well, the purpose of these cells is really to detect oxygen and carbon dioxide levels in the bloodstream and get that information back to the brain. Well, for unknown reasons, mutations can occur within these paraganglia cells and causing tumor formation. Most and almost all paraganglia are usually benign. There is a malignant variety of paraganglioma, and it's just referred to as malignant paraganglioma that can metastasize, that can spread from the ear to lymph nodes regionally and throughout the body. So it's important to make the diagnosis. Typically, the diagnosis is made by an astute physician who encounters a patient who has pulsating tinnitus and hearing loss, and a conductive hearing loss. And when they look inside the ear canal under a microscope, they can visualize the tympanic membrane, and they can actually see the paraganglioma behind the eardrum. This is what we call a glomus tympanicum. These paragangliomas also have another name, and that is glomus tumors. So these glomus tumors that arise in the middle ear are called glomus tympanicum tumors. Those that arise in the jugular bulb are called glomus jugulare tumors. These are just a difference in names, if you will, but really the same kind of tumor. A glomus tympanicum tumor, very easily treated with removal and excision. A glomus jugulare tumor is much more problematic, and treatment is really tailored according
Type of tumor manifests mutations genes related to hypoxia, including the PDAS1 gene. Prolonged exposure to hypoxia is described as an abnormal risk factor for the development of paraganglioma in individuals living on high altitudes. Chronic hypoxia in patients with congenital cyanotic heart disease. Heart defects present at birth involving low levels of oxygen in the blood also poses an increased risk for developing this type of tumor.
Male presenting for evaluation of hoarseness, arising along the posteromedial aspect of the right carotid sheath between the right carotid arterial system superior to the bifurcation and the right internal jugular vein. There is a circumscribed T1 isointense mass with avid heterogeneous post-contrast enhancement and mild diffusion restriction. The lesion was hyperintense on the T2 steroided images, and several prominent flow voids were identified. The lesion splays the carotid and jugular vessels. The differential includes right glomus vagale paraganglioma, vagal schwannoma, neurofibroma, and carotid space meningioma. This is a right-sided glomus vagale paraganglioma. Glomus vagale are avidly enhancing lesions in the nasopharyngeal carotid space arising approximately 2 centimeters below the level of the jugular foramen. Glomus vagale displace the carotid anteromedially, while the jugular vein is displaced posterolaterally. The parapharyngeal fat plane is effaced anterolaterally, and the styloid process is displaced laterally. Multiple lesions can be seen in the setting of hereditary paraganglioma and pheochromocytoma syndrome, multiple endocrine neoplasia type 2, and von Hippel-Lindau syndrome.
Application in medicine are catecholamines creating tumors of chromatin cells with frequency germinal, somatic, or prosthetic mutation is genes that are involved in hypoxia-related patients, include BHL, and those mutations result in aberrant and uncontrolled activity of hypoxia-inducible factors, even under normal levels of oxygen, a condition known as pseudohypoxia.
One, because we have ventricle, the pulmonary artery is usually attached to the right ventricle. In this case, they're switched. And this is a very interesting disease because it results in two separate circulations of blood. The right side of the heart is pumping through the systemic circulation, and that's like an isolated system from the left side of the heart, which is pumping through the pulmonary circulation. So in order for somebody with transposition of the great arteries to survive, they need at least one shunt. This could be an atrial septal defect, a ventricular septal defect, or a PDA, a patent ductus arteriosus, which is a connection between the aortic or the aorta and the pulmonary artery. So they need some kind of connection between left and right heart to allow for mixing of the blood, to allow for them to survive. In that image that we have on the right there, we see an atrial septal defect. That's allowing for the mixing of the blood, turning it purple to pump through the systemic circulation, allowing for the patient to survive. Next, we have 3. 3 helps us remember tricuspid atresia. There's not much to do with the 3 here, except for the fact that it has to do with the tricuspid valve, except we don't have a tricuspid valve. Instead of a valve here, we have a wall. Tricuspid atresia is the tricuspid valve being replaced with a solid wall that you cannot get past. So this means that in order for the blood to complete a circuit, you have to have both an atrial septal defect and a ventricular septal defect. Blood needs to be able to flow into the right atrium from the superior vena cava, through to the left atrium through an atrial septal defect, down into the left ventricle, and then back to the right ventricle through a ventricular septal defect. We need both septal defects in order for somebody with a wall replacing the tricuspid valve to survive. This is tricuspid atresia. 4 helps us remember tetralogy of Fallot. Tetralogy means 4 things. We have 4 stenosis. The pulmonic valve is thin. It's hard for blood to get through. We're going to skip over to D because I see it kind of as a direct result of A. D is right ventricular hypertrophy. If we're not easily pumping blood through the pulmonic valve, if we have pulmonic stenosis, A, and we cannot pump blood through it, the right ventricle is going to get much larger as it struggles to pump blood. This is a compensatory mechanism, right ventricular hypertrophy, as a result of pulmonic stenosis. So that explains A and D. Next we have B. B is a overriding aorta, meaning that the aorta, which usually kind of points to the left ventricle, is pointing into both chambers here. The aorta is taking blood from both the left and the right ventricle. Normally it just takes blood from the left ventricle. And kind of going along the same lines as the overriding aorta is C, which is a ventricular septal defect, which essentially kind of connects the two ventricles. We don't have much of a septum between the left and the right ventricle, which allows the blood to mix in between the ventricles and go mostly into that overriding aorta. So these 4 qualities contribute to the
Hypoxia has been reported and some body risk factors for Paragangliomas and Paragangliomas that arise in persons living at high altitude. We previously found that patients who have chronic hypoxemia, that is due to cyanotic congenital heart disease, were at increased risks for pheochromocytomas and paragangliomas, but the mechanism understanding these associations is unclear. Pros and cons. Pros to today, there is no medication that can effectively treat the paraganglioma by inhibiting the HIF-2 alpha protein. Now, following the results, Dr. Rodrigo A. Toledo, together with his colleagues from UTHSCSA and Anderson Cancer Center of the University of Texas are exploring the possibility of using HIF-2 alpha clinically as a molecular target deploying the first blockers. Cons. Correlation or complete interaction of the aorta, Leucine anomaly, hypoplastic left heart syndrome, tetralogy of Fallot, Total anomalous pulmonary venues return to inspection of the great arteries, truncus arteriosus.
The problem for me regarding this question is that remembering all 5 of the congenital cyanotic heart diseases is a pain in the ass. Luckily, there is a mnemonic to remember all 5. The first thing to remember about the cyanotic congenital heart diseases is that they all begin with the letter T. The next thing to remember is that there are only 5 of these cyanotic congenital heart diseases. The mnemonic follows the numbers 1, 2, 3, 4, and 5. Number 1 represents the great vessels joining to make one outflow tract in truncus arteriosus. Number 2 represents the major vessels being switched in transposition of the great vessels. Number 3 stands for tricuspid, for tricuspid atresia. And number 4 stands for 4 defects in tetralogy of Fallot. Lastly, 5 stands for 5 letters for the acronym TAPVR, standing for Total Anomalous Pulmonary Vascular Return.
In Ecuador, here we report the identification of gain of function somatic induction of EPA-S1 which uncut for HIF2 alpha in Paragangliomas and Paragangliomas in 4 of 5 patients, 80% who represents with cyanotic congenital heart disease. The clinical and genetic details of the patients are shown in Table 1 and in the supplementary appendix. The affected results are known to regulate height to alpha stability and when done results in constitute height to alpha activation and tumor growth in vivo.
Comes in, typically it goes straight to payroll, specifically to the inner mitochondrial membrane where oxidative phosphorylation takes place. Oxygen is used in one of the last steps and serves as an electron acceptor, and this allows the process to finish and produce ATP. So without oxygen, we can't finish oxidative phosphorylation and produce ATP. But why does the whole factory fall apart when payroll stops making ATP? Why don't they just pause for a bit or take a little break? Well, when certain workers stop doing their jobs, things get a little out of hand. One super important worker is the sodium-potassium pump on the cell's membrane. Pretty much like the bouncer that makes sure there isn't too much sodium diffusing into the cell, basically by pub, or bulge outward from all this water. And this is a sign that the cell's cytoskeleton, or this structural framework, is beginning to fail, and is letting water slip through. Finally, the rough endoplasmic reticulum, or the rough ER, also swells when the cell swells. And remember and stop making proteins. So protein product called lactic acid, which lowers the pH inside the cell. This more acidic environment can denature or essentially destroy proteins and enzymes. Now, up to this point, it's not all bad, because one super important thing about these processes that happen to the cell is that they're potentially reversible, meaning that if we all of a sudden get oxygen again and start making ATP, that keeps the cell together. Also, endonucleases can be activated, which can cut up DNA, the cell's genetic material. And if we get back to the lactic acid, as more lactic acid builds up and the environment gets more acidic, the lysosomal membrane can be damaged as well, which usually houses these hydrolytic enzymes whose job is...
Conclusion. This observation, along with the existence of a clear link between Paragangliomas and Paragangliomas with mutations that lead to the activation of HIF-S and cellular hypoxia suggests an increased sensitivity of chromatin cells to HIF2 alpha mediator. It is possible that the EPA's mutation endochromatin cells that have been exposed to chronic hypoxia hypoxia with the ability to amplify the oncogenic proteins of high 2 alpha.